Welcome back, everybody. This is going to be our review on chapter 16, which dealt with specific immunity, acquired immunity, whatever you want to call it. So remember, all lymphocytes will be starting in the bone marrow. The B cells will be maturing there, and then they'll move into the periphery and hang out in the lymph nodes, while T cells are going to mature in the thymus before stationing in the lymph nodes themselves. So they'll also be circulating out in the periphery too. They won't just stay in the lymph nodes, by the way. So lymphocytes are white cells that are, agranulocytes. These are going to be our B cells and T cells. Anytime they're activated, they're going to proliferate first, and then they're going to differentiate. So first they multiply, then they turn into whatever kind of cell they're going to be. All of our specific immunity lymphocytes are going to go through clonal selection, and basically that is removing any of the clones that were made during a maturation in the bone marrow. We're going to remove those guys, the ones that would be reactive with self. So the ones that would have otherwise seen self as foreign when it shouldn't be doing that, right? That happens during clonal selection. All right, antigens are anything that can provoke an immune response, and anything that has provoked an immune response, we're referring to as an immunogen. You can use those terms interchangeably, by the way. Then, so for anything to be an antigen, it needs to be foreign. Of course, it has to be a certain size. It has to be, accessible and all that sort of stuff in order for us to be able to respond to that antigen. So haptens are things that are too small for the, B cell receptors, for example, that variable region to interact with. So they have to have a carrier to help them so that they can react appropriately. This is the case with the tetanus toxoid vaccine. We couldn't react to the tetanus toxin without it being along with other molecules. Super antigens are any antigens that will induce overreaction by the immune system, which can be deadly. We see this in the case of toxic shock syndrome, right? And where basically the body is going to stimulate so much of an inflammatory response and send out so many cytokines, which are our call to arms, chemicals, that they're going to have a cytokine storm, and that can react with massive amounts of inflammation all throughout the body, leading to shock and leading potentially to death. Antigen presenting cells are going to be our macrophages, our dendritic cells, and our B cells, according to the lecture slides. And we also are including the monocytes, which are our precursors to our macrophages. Remember, they're going to be presenting things on MHC1 or MHC2. All cells, including our antigen-presenting cells, have MHC1. This is what's going to be where we're going to present anything foreign from within a cell. So that's endogenous antigen. MHC2 is only going to be possessed by those antigen-presenting cells. They're going to go around, find something outside of the cell, exogenous antigen, digest it down into parts, and present it on the MHC2 molecule. So that brings us to the T cells. And of course, the first T cells you would want to talk about would be the T helper cells. Whenever you have antigen bound with MHC1, which were those endogenous antigens, the first ones that we just talked about, the CD4 positive T cells that react to those will differentiate as Th1 helper cells. So that's because they interacted with MHC1. These guys will be involved in interacting in activating the CD8 cytotoxic T cell response. Both the Th1 and the Th2 CD4 T cells are going to activate the macrophage response to allow macrophages to go back to the field and start, killing off and ingesting more bad guys. Remember now, our MHC1 is going to present our endogenous antigen from within the cell. It will activate our CD4 positive T cell to become the Th1, the T helper 1 kind of CD4 positive T cell. And those will be involved in the activation of the CD8 positive cytotoxic T cells. And they are going to kill, they're cytotoxic, which means they're going to kill target cells using their perforins and granzymes, sorry. Perforins are going to poke holes in the cells. The granzymes are going to be like digestive enzymes that break down stuff within the cell. So that will cause the cell to start breaking down on the inside, start leaking out its content, and then eventually undergo apoptosis, which is cell death. Antigen and MHC2 together, remember that's going to be our exogenous antigen stuff that came from outside the cell. We're going to activate our CD4 positive T cells, and they'll become Th2, T helper 2 cells. These are involved in activating the macrophages again, but they're also involved in activating the B cell response. And B cells that, again, we're going to see that in a moment, but B cells that are activated will then become plasma cells, and plasma cells are heavy antibody producers, so producing a lot of antibody in response to these things. So that's important for activating that. Then we also, of course, have regulatory T cells, or other T cells too, but these are the big ones. All right, our B cells. B cells, when they have been activated and differentiated, they will differentiate into their effector form, which is the one that does the job, which happens to be called plasma cells. Plasma cells are the B cells that have differentiated and are now producing antibodies, secreting it into the environment. FAB, all right, so we talked about the immunoglobulin. It's Y-shaped. It has, I don't have the thing now. Here it is. It's Y-shaped, has a heavy chain, and it has a light chain, so we'll draw it out here. Heavy chains are shaped like this. Light chains are the shorter versions up here. This is where the actual antigen binding is going to be happening. It's actually literally going to be binding with the epitope, which is the specific part of the antigen that this antibody would be reacting to. And this is the constant region. The constant region. So this part is kind of what gives that antibody its purpose, like what it would be doing for the cell, because they can have different purposes. So that's the basics of that. So the FAB is that variable region. So it's going to be everything up here. That's the FAB. So like the antibody, FAB, fragment AB. That's going to have the variable region. The FC is the constant region. All right, antibody, what it does, it will neutralize, opsonize, coating, agglutination, complement, and antitoxin. So neutralizing will be essentially covering something up with a whole, coating something really, but covering an organism up with why can't I think? Okay, neutralization. We're going to have our antibody binding onto like sites on the organism that allow it to bind to our cells. So the docking mechanisms essentially, or with sometimes with enzymes that are associated with and that. So we'll bind with it so that those things can't happen. Opsonization. We're going to be coating something with antibodies so that's easier for phagocytosis to occur. Coating in general is just going to be coating something in antibiotics so it can't antibody so that it can't do the thing it's trying to do. Complement activation, that's classical, the classical route of complement activation. Complement activation this way, we have antibody that's going to signal for cleavage of all those complement proteins and get that cascade moving so that we can develop that membrane attack complex that pokes holes in the enemy cells. And also we can have antitoxin effects from the antibody whenever the antibody is associating with an actual toxin and keeping it from being able to do what toxins do. So, all right, the types of antibody that we have, the main ones that we are involving in actual immune response. We talked about IgG, that's our workhorse antibody. We have IgM, that's our pentamer. So it has 5 of those Ys altogether. That's our initial antibody. But it's not as effective as the IgG. Then we have IgA. This is a pretty effective one. This is involved with mucous membranes. IgE was involved with allergies, which we'll talk about in the next chapter, I believe. And then IgD, that is just a receptor, the B cell receptor. Okay, so our antibody response, we start off always with a primary response. This is going to be the latent period with IgM at first made, and then we will switch to class switching to IgG later on. That'll help clear out whatever we're fighting, and then whatever we're done, and we wait a couple years and you're re-exposed, and then you'll have your secondary or amnesiatic response. This is going to be IgG right off the bat, right away. Be a much greater response and often be so fast you don't even know you were exposed to the thing. Right, there's different kinds of immunity that we talked about. Natural would be anything that you got not from a hospital, normal exposure to a thing. Artificial is going to be anything from medically induced or introduced. And then we have active versus passive. Active is going to be the things where you're responding with your own immune system to create antibodies and therefore even a memory response. Whereas passive is we're providing antibody from somebody else to you to help you survive the infection. So using those antibodies. All right, so natural active example would be responding to an illness like chickenpox. I had chickenpox. Natural passive would be breastfeeding, the mother to the child, antibody transfer. Artificial active would be vaccination, and then artificial passive gamma globulin infusions, which we talked about in the other lecture. Okay, so vaccines, we can have live attenuated vaccines, we can have killed vaccines. We talked about the viruses that were cleared out and filled with mRNA, or the little, after that we had the lipid balls or whatever that we filled with mRNA. So the virus one that had the mRNA, that was the COVID Johnson & Johnson. And then the lipid layer that was surrounding the mRNA, the genome essentially of COVID, that one was Moderna and Pfizer. So, okay, so yes, we'll talk about those. Subunit vaccines, that just can be part of taking apart like just the spike protein of a virus. For example, toxoid, which we've already mentioned with tetanus toxin, right? So it's toxoid vaccine. Conjugated, which means that we're putting two things together, that together they elicit a much stronger response, and therefore it's more effective immune protection. And then we can introduce those to the body either subcutaneously, intramuscularly, or oral nasal route. We also have the intradermal. I'm not as worried about that. Okay, remember that every time that you get a vaccine, that we are using the exact same immune system that you would have been using if you had actually gotten sick with the thing. It's just, hopefully there's no infectious agent here and you can't get sick from this and die potentially. People don't realize that you can die even from the measles, and you might say, oh, not that common, but you'd be surprised. Yeah, and we didn't really talk about herd immunity that much in this chapter, now that I think about it, but remember that herd immunity really is going to be talking about how many people have to be vaccinated in a population in order to provide protection to the ones that can't be vaccinated. And so we did talk about that in chapter 14, I believe, in our epidemiology chapter. So if you guys want to go back and refresh your memories on that, remember that this is relating quite directly back to that. And then we talked about side effects and misinformation. I mean, this part was pretty straightforward. Remember that side effects sometimes are a sign that your vaccine is working and not a sign that you're sick. They're not necessarily a bad thing. Other times they can be a bad thing, can be dangerous. So of course, look out for yourselves. Make the judgment based on what you and your doctor would say. And then, of course, the misinformation, one that we talked about, of vaccines causing disease, just regular vaccines causing disease. Apparently, people thought the flu one did. It's not true. And then, of course, the mercury in the MMR vaccines is not a thing. That's not a thing, and the paper has been retracted. So that's not a thing. So that is that. And that is the review for chapter 16, and I'll see you guys for the next one.